All right, so I'm going to try to help you with this protein synthesis worksheet that I gave you. So a lot of the stuff we've gone over. Remember the tenets of it. So if we have a eukaryotic cell over here, remember that transcription is going to occur in this nucleus region here, right? And it's going to make pre-mRNA, which is going to be edited to make mRNA when we remove the introns. We add the G cap on the 5' end and the poly A tail on the 3' end. And we remove those introns, like I said. And then those mRNA, right, are going to exit out of nuclear pores, right? And they're going to find ribosomes. So they're going to find ribosomes in the cytoplasm, and those ribosomes are going to start translation. And then the translation is going to occur as the ribosomes, most of the time, will find the rough endoplasmic reticulum or the rough ER and attach there and then start to make proteins. All right, so I've kind of explained that here. So remember transcription, translation, where they occur. On the DNA, nitrogenous bases, remember they're read by RNA polymerase, one at a time, right? And then they make codons, and codons are going to be those sections of 3 amino acids, or not 3 amino acids, but 3 nucleotides at a time, right? Remember the tRNA is going to have the complement to the codon on the mRNA called the anticodon, right? And then I put a bunch of amino acids together. Ultimately, they're going to make polypeptides. So we get to part C, which some of you said that you wanted help with. So there's some strategies here. Remember that amino acids always start at a start codon, right? So my advice to you is then find all your start codons, right? TAC on the template strand is always going to be your start codon. So we can come down here and I can just start labeling the TACs, and that's where my starts are going to be, right? My stop codons, right? So my stop codons, remember if we have our codon chart here, I got UAA here, I got UAG, I got UGA, right? So if I was going to write those as template strands, I would have ATT, right? I would have ATC, and I would have ACT, right? So those are what I'm going to look for over here in my template strand. And when I find them in my template strand, I'll write a stop on them, right? So I come over here and I'm looking, and I got to stop all the way here at the end. Right, my ATC is all the way here at the end. So that's going to be my stop. Right? All right, so I come over here to the next one. Right? I'm searching around. I got an ACT here, so that's going to be my stop. All right, we come to my next one. I got my ATC, so that's going to be my stop. So find your starts and your stops, and that would be my advice to you. And then after that, right, I can start one at a time filling in my amino acids. So mRNA, let me pull my paper I've already done. Oops, on this one up here, I had an ATT here. So that was my stop there. I don't have to come all the way down to here. All right, I missed that stop there, sorry. So if I was going to make my amino acid, my mRNA now, right, so I have Well, it's going to be hard to write, so let me erase this. I'll try to make it a little bigger so I can write my codon stretches, or I can read them. All right. So I've got AUG, I've got UGU, I've got GCC, I've got UCC, I've got CU. Oh, sorry, not CU. CAU. I've got GCG. I've got AU. I've got AG. I've got U. All right, so I had U, AC, and then this one right here we said, we'll stop. Right, so then I can go over to my my codon, my amino acid chart, right? And I can look at the first one. I say A, U, G, right? So that's right here, A, U, G. So my first one is going to be MET. And I go to my next codon was UGU. So I come over here to UGU, and I have right here, and that's CYS. Then I go to my next one was GCC. I got G, I come over to C, and I got GG. GCC there. So I'll say that's ALA. My next one was UCC. Again, I go U, I come over to C, I got UCC here. That's serine. I got CAU. I start at C, I come over to A, I find CAU. That's histine. I got GCG, GCG, I'm down here. I have another ALA. I got AUA. I come over here to U, I got AUA, I got another LLE. And then my last one, UAC. I got UAC, I got TYR. Remember, there is no amino acid for a stop. So then I would stop there. So that's my protein strand right there. It's always good if you want, you can put a little dash in between each one of them. So I know where your amino acids are. Okay, so that's essentially how you do those. Again, find your starts and your stops, and then fill in all the amino acid up to your stop. Don't include an amino acid for your stop. All right, so the true and false, or the circle, the correct answer. We've gone through all of these, so review your notes. And a lot of this is kind of a review of what we did in the first page. All right, so fill this in here. We have DNA. Remember, DNA runs 3' to 5', right? Because that's my template strand. My mRNA runs 5' to 3'. My tRNA then is going to run 3' to 5' because they're all complementary of each other. All right. So my mRNA, right, is the complement. Remember, I don't have T's, so I'd say U, GU. All right. So my next amino acids would be A's become U's, CU, and then I've got GCC, and opposite of T is A, UG, and I've got AC, U. So my tRNA would be anti-complementary. So it's the same as the DNA essentially. So ACA, AGA, CGG. Remember, there's no T's, so this would be U, AC, and then I would have a U, CA. When we came over and did our amino acid, let me make this a little smaller so I can fit it. When we would come over and do our amino acid, remember amino acids are based on the codons on the mRNA and not the tRNA. So for the first one, I wouldn't look at ACA, I would look at UGU. I'd come over here, U, G, and then U, right? Which would be right here. And that would be a cytosine, or cysteines. So that would be CYS. And then I have UCU. I'd come over here. I get UCU right here, and I would have serine. Right? I've got GCC, G, and I come over to C, and I got GCC right here. That would be ALA. And I've got AUG, and we all know AUG. Now, we can have methionines. It doesn't have to be the start codon. Right, so MET. And then I've got ACU, A over here to C, and then U. And that's threonine. I got THR. Right, so that's how you did that. If I come down to the rest of it, most of this information we've already talked about. It's already been covered in this sheet. The end we haven't talked about yet. So if you pause this, here's all the information to fill out the end of it, because we really haven't got to that yet, but I will be talking about this next week, right? So if you want to go ahead and pause this and go ahead and write all those answers down, that would be fine. All right, I hope this helps you out, and I'll talk to you soon. Stay safe, stay well, and I'll see you next week.